this, the GFP expression in the mesenchymal stem cells without gene jammer and with gene jammer. And we did do flow on this as well to show this. When we did use the gene jammer, we were concerned again about the possibility of detrimental effects and toxic effects on the cells. We did not see any obvious toxicity problems when we looked at PI positive cells. So these were not dead cells that we were seeing the GFP in. We did a number of experiments over time looking at the effect of the adenoviral dose on the transduction efficiency. So increasing adenoviral concentrations, obviously without the gene jammer, you saw a steady increase in GFP positive cells, but again we plateaued fairly early with the gene jammer. So this is obviously important that you can use lower doses of the adenoviral vector and get still very high transduction efficiencies. So again, this was just relative fluorescence units. Again, some indication of the level of or number of viral particles entering the cells, and remain fairly constant without the gene jammer, but at the very high end of gene jammer, you can really load up the cells with the vector. And again, we did not see any really statistically significant detrimental effects when using the gene jammer. When we're very interested in because different culture conditions can affect the differentiation of these mesenchymal stem cells, we're really interested in looking at the effect of fetal bovine serum concentration on the transduction of the mesenchymal stem cells. And even at 0% fetal bovine serum, we still saw a relatively high level of GFP expression, positive cells, and we could also go up all the way to 20% fetal bovine serum and still get good transduction with these vectors. So from 0 to 20%, we're still relatively uniform in the transduction efficiency. So we're quite happy with those results as well, because again, if you're going to use this in a cell that you're going to look at differentiating them later, you know, if you want to use with different concentrations of fetal bovine serum, or hopefully without fetal bovine serum in a more defined system. The next experiment, we looked at transduction of the mesenchymal stem cells, and now this just with with just the 85 alone fiber. So again, we moved back to just the more primitive type of adenoviral vector, and again, it did not seem to matter in the gene jammer transduction that whether it was the 85 or the 85F35 vector, we still saw very high levels of GFP positive cells with that, and again, no detrimental effects on viability of the cells. And again, you can see the different amounts of GFP expression in those cells. Okay, we did look at lipofectamine as a transduction efficiency as well, and yes, we saw some beneficial effects of the lipofectamine very similar to the gene jammer in many ways. However, we saw with lipofectamine there was some detrimental effects on cell viability with using some of these concentrations. So higher ends of lipofectamine, we did see some problems with the viability of the cells. When we looked at differentiation of these cells transfected with the GFP, we did see that the cells, again, could differentiate even though they had the adenoviral vector transduced into them, which isn't surprising. And so, again, with the oil red, we saw positive cells as well. So these cells could differentiate again in the chondrogenic and osteogenic pathways even after transduction with these viral vectors. And at relatively high rates, again, which was very encouraging as we move forward with this work. So with the first study of the conclusions, the porcine mesenchymal stem cells are poorly transduced with just the adenoviral vectors, but the gene jammer transfection had a marked increase in transduction without any toxic effects. And it's not restricted to one type of vector. It had similar results with the 85 and the 85F35 vectors. Even with the addition of approximately 10% serum during transduction, there was some improved efficiency, but again, the main thing was that you could vary the levels of serum and still get good rates. And they do differentiate into the multiple pathways. So the second study, we were looking at the effect of gene jammer, not on pig mesenchymal stem cells, but this time with human mesenchymal stem cells in rodent cell lines. And objective of this, and this was done in collaboration with a group at Baylor Medical School, to investigate the effects of gene jammer on the efficiency of human mesenchymal stem cells with adenovirus containing GFP. Again, as if the gene jammer's positive effect on transduction was dependent on the presence of the receptors. And we believe that it was an independent method, but we needed to check that out for sure. And then to investigate the effects of gene jammer on the transduction efficiency of human and murine cell lines with BMP2. So similar in many ways, we looked at different viral particle loading of the cells, and again we saw very similar effects with the human mesenchymal stem cells that without the gene jammer is fairly low at the low concentrations. If you obviously, as you increase viral particle concentration, you get a higher transduction efficiency, but again, you were able to get, actually this is reversed, sorry. The gene jammer is in the white bars, and the control is in the dark. So we did see very similar results as we had seen with the pig cells as well. And you obviously, if you look at flow cytometry for quantification, this backed up the other results we see. So the next experiment in this group of studies, we're asking, does gene jammer enhance uptake through adenoviral receptors? And so looking at various cell lines with different receptor populations, the A549 human mesenchymal stem cells, CHO cells. So, and we looked at different viral vectors and with and without gene jammer. And the endpoint was, again, flow flow to look at the percentage of cells. So if you look at the 549 cells, high levels of the CAR receptor, the integrin, as well as the CD46, whereas you see with human mesenchymal stem cells or bone marrow derived mesenchymal stem cells, very low levels of the CAR receptor, higher levels of the integrin and CD46, and it shows cells as low all the way across the board. So we had a varied number of cells with different characteristics as far as their cell surface receptors. With the 549 cells, there was no difference between the two vectors, or there was no difference between the control or gene jammer and the number of GFP positive cells. Again, this is expected as the 8049 cells have high levels of receptors, so the control would be very high. The human mesenchymal stem cells, again, we saw differences, again, with the gene jammer addition, and probably because of it being a receptor-independent pathway. And then as well with the CHO cells, we saw differences between the gene jammer and control on both the viral vectors. So again, this suggests in our mind that this is a receptor-independent means of introducing the adenoviral vectors into the cells. So getting on to what might be some effective gene therapy, we were looking at using the 85 expressing BMP2 and using the control, again, without gene jammer and with gene jammer in the human mesenchymal stem cells or the osteoblastic cell lines. That was over a 42-hour period where they were exposed to supernate and then was quantified for BMP2. There was a cellular AP activity assay as well that was performed, and then the cells were also injected into non-skid mice for the bone assay over a 2-week period. So again, you can see BMP2 production with low viral particles per cell. Again, the gene jammer had a significant effect on the expression of BMP2 because of the high transduction efficiency with delivering it. Looking at BMP2 activity, we saw similar results again as the viral particles were increased with the 85 vectors. And so again, suggesting that this is an efficient way of introducing a vector into the animal for gene therapy. And in the bone formation over 2 weeks, you can see in the controls where the cells were injected without the gene jammer, that you saw no bone formation. But as you can see, when you have the gene jammer added, you can see the bone formation in the muscle of the limbs of those mice. So with the mirroring cell lines, again, we saw a similar effect with the BMP2 activity. It's a bit washed out again, but again, we saw bone formation in the gene jammer animals as well. So with the mirroring cell lines, we are also effective. So the conclusions that we have from this study is that gene jammer can significantly enhance adenal entry into the cells, and this can be obviously for human therapeutic, human mesenchymal stem cells. It's It's possible with those cells. The positive effect of gene jammer on adenoviral transduction is independent of the fiber type 5. Again, this is a receptor-independent method of introducing the adenoviral vectors. And the results suggest that gene jammer stimulates viral entry with a mechanism independent of the receptors. The presence of gene jammer during infection the cells with the adenoviral vectors increase the expression of a functional protein, that being BMP2. So the work was done again by Pablo Bosch, as well as our collaborators Alan and Betsy Davis at Baylor Medical School. Scott Pratt was involved in this work with a company that funded this work as Biogen, and that's a company out of Austin, Texas. And with that, I'd be glad to answer any questions.
Thanks, Steve. Open the questions. Thank you. Do we have someone with microphones? Here it comes.
That's a very nice series of studies. I have two questions, please. One is, why did you choose adenovirus, and you wanted only expression for a period of time rather than constant with AAV? And my next question, if I put it together, is that's an interesting way of doing it, because what do you think? You mentioned earlier that you can influence the cell division, and a very attractive agent, for example, is HTERT, which could enhance the cellular renewal in a terrific fashion. But we were very interested in that, and then we had about one paper showing that it actually produced malignant transformation. And probably after many times or many divisions, and your strategy might get over that.
Yeah.
Your thoughts, please. Why did you use one? I mean, the EMP2. Why do you want it for only a period of time?
Well, this is really a proof of concept type of study, and I didn't show the data on the AAV vectors that were used with GFP, but they were positive. I mean, very similar to what we saw with the 85 and 80 and F35. So we could, with the gene jammer, get very high rates of transduction as well as stable integration. So we could select with the AAV, we could do flow cytometry and I mean, fax and sort out the cells and do clonal derivation of cell lines with that. And I just didn't have enough time to show you that, but it works just the same across the board for any of those. And obviously, if we want stable transfection, that's the way to go. You know, there are people, and you know better than I do, that are suggesting that there may be some things that you'd want short-term expression, and that's why you would use that, obviously. But I've also had questions, well, does it also work with lentiviral vectors or other vector systems? And we just haven't tried that as yet. But for BMP2, that's the vector that the people at Baylor had available and were interested in. I don't know. It's a good question. I should ask if they had the AAV version of that, and probably be worth trying as well. But the AAV does work with gene jammer, and you can get clonal selection of those cells.
HTERT?
HTERT. Yeah, that would be of interest for expanding the cells over time. Trying to think. there have been a few groups that have put in H-TERT into cells and fibroblast cells, and yes, they work to some extent. I know that a group that's, I'm trying to remember the name, in North Carolina, that they don't believe that H-TERT alone will be sufficient to get long-term expansion of cells. That you probably need some other things like a large T antigen or something like that you'd transiently be able to express from time to time. That would be useful. So, but I think, yes, H-TERT would be a really nice one to put in and
Can I
expand.
ask another question?
Please.
Bad publicity about adenovirus. Is that going to last with us, or was it a dose effect, or
Yeah.
have we seen the end of it?
I'm not I'm more of the stem cell person than and so anybody else that would like to have their point of view put across, I don't know.